Eastern fire salamander, Salamandra infraimmaculata. Professor Gad Dagani, scientific editor. Amir Marshansky, photograph and films. Sharon Zamir, film editor. Urodela order, salamanders and newts, which comprises 9 families, with more than 550 species. And the Apoda order, comprising 3 families, with more than 170 species, mostly in the family of Sicilian. The latter order is the least studied of the amphibians. In Israel too are Urodela, banded newt, Trichurus vittatus synonym Amatotriton vittatus, and fire salamander, Salamandra infraimmaculata, and tree frog, Hyla savignii, green toad, Bufo viridis synonym Sedepidalia viridis, Rana badriagi, synonym Rana ridibunda. Spadefoot, Helibate syriacus, and hula-painted frog, Latonia nigravinder, belonging to the Elytidae family. The Salamandridae family, comprising 16 genera and 66 recognized species, represents one of the most diverse groups of extant salamanders. Salamandra genus have the largest geographic distribution of any salamander family, extending across the Holarctic continents of Asia, Europe, and North America, with a small and recent expansion into North Africa. The Salamandra genus includes 6 species. Some are defined as subspecies in a wide distribution in Europe, Northern Africa, and Asia, including Asia Minor and Israel. This division and the large variation between populations sometimes make it difficult to determine the species or subspecies with certainty. Steinfartz et al. Suggested classifying the species in Europe into 6 groups according to research on mitochondrial DNA. Salamandra, Salamandra infraimmaculata, Salamandra corsica, Salamandra atra, Salamandra lanzi, and Salamandra algira. Accordingly, the fire salamander, Salamandra infraimmaculata, is located in Israel and the Salamandra algira species in northern Africa. Karahizer et al. Studied the differences between the fire salamander in Israel and in Turkey, where there is also a species of salamandra. The southern distribution border of the salamander genus is northern Africa and Israel. In Israel, there are 3 main areas of habitats where salamanders live: Mount Carmel, the Upper Galilee, and Tel Dan and the slopes of Mount Hermon. Another possible classification is the areas of the Hermon and the northern Golan Heights, the Naphtali Mountains, Marim Hagalil, the western Galilee, the Tefan area, and Misgath. Tel Dan population is 190 m, where the environment conditions of this habitats is different from the others. Tel Dan stream. Fast-flowing stream. Temperature range 15 C to 17 C, depth 5 to 100 centimeters. The habitat in the Galilee, the Meron region. This region includes the highest mountain in western Israel, Mount Meron, whose altitude reaches 1208 meters. The altitude and unique location of the region create diverse conditions that affect vegetation and animal life. The characteristic vegetation is primarily oaks and pistacia of a number of species. This area of Mount Meron is relatively rainy compared to other parts of Israel. The average annual precipitation is 1,000 meters of rain. The summer is relatively comfortable, except for a number of days of heat waves a year. In the Galilee, there are areas with dense wildwood and exposed areas. The salamanders are found in locations that provide the possibility of breeding around water sources, but also in areas at a distance from them. The habitat in the Carmel. With its high altitude, the Carmel is a complex habitat built from 2 main ranges. A prominent phenomenon that occurs there is karstic melting. The upper range is composed of chalkstone from the Cenomanian age, and the altitude is about 500 meters. The peak is around Osphia at an altitude of 548 meters. The second range, parallel to the first, is composed of chalkstone and marlstone from the early Cenomanian age. This range begins in Tirat Hakermel, continues through Beit Oren, and reaches daily at El Carmel, and is crossed by a number of wadis of seasonal streams. Its altitude is 450 meters. And the peak is Mount Shokef at an altitude of 497 meters above sea level. The yellow spot pattern of salamander in various habitats at the southern border of its distribution in Israel. Different dispersion of spots on the back of the salamanders in the Dan, Sasa, and Yayam populations. The patron that appeared on the salamander body is, in 1 row, 2 rows, or scat-tiered. The size and shape of the spots differed among the individuals belonging to the various populations. Some were round and others had different shapes. Comparison of the patterns of yellow spots on the backs of the salamanders 1 row, 2 rows, and scattered between habitats Dan, Sasa, and Yayam. Differences were not significant. The pattern of spots in salamanders from the Tel Dan population. The pattern of spots in salamanders from the Sasa population. The pattern of spots in salamanders from the Yayam population. Comparison of percentage of yellow spots on the black back of salamanders in the different habitats. No significant differences were found among the Yayam and Sasa populations. Both populations differed from Dan salamanders. The formula for calculated the percent yellow of color is percent of yellow equals A x 100A plus B. A is the area of the yellow spots and B is the area of the black color. Number of spots on the heads of salamanders from the different populations. Color patterns in the various subspecies of S. Salamandra. Some of these patterns also appeared in populations of S. Infraimmaculata. Spot patterns of S. Salamandra that did not appear in S. Infraimmaculata in the present study. And spot patterns of S. Salamandra that also appeared in this study in S. Infraimmaculata. The skin mucus of the Near Eastern fire salamander. All species of the genus Salamandra secrete poisons, termed Salamandra skin poison, which serves to protect them in conjunction with the salamander's warning colors. Metabolites were determined in skin mucus of the Near Eastern fire salamander by LC-MS analysis. The salamanders were sampled from 3 different sites at the southern border of their distribution in Israel. Kibbutz in the Upper Galilee and in the Lower Galilee, both semi-arid mountain areas, and Dan River, where water is available year-round at a constant temperature. In total, 1112 metabolites were detected. Venn diagram is shown in a list of 14 metabolites that were found in the 3 populations Manoff, Sasa, and Dan. In total, about 1,112 different metabolites were found in the mucus secreted from the salamander's body. Many of the metabolites found in the mucus were known SSPs. 891 metabolites were common to the 3S. Infraimmaculata populations, 119 were unique to the Manoff population, 84 to Sasa, and 7 to Dan. The percentage of metabolites shared between the salamanders from Dan and Manoff, and between those from Dan and Sasa, was significantly lower than that between the 2 mountain populations. No significant differences in normalized area were found for most of these metabolites among the different populations of salamanders. Except for cholic acid and dihydronandrolone between Sasa and Dan, and samonine between Manoff and Dan. Samonine and samanderone were present at different levels in the mucus of the 3 populations of salamanders from different habitats. Both were higher in the Manoff population than in the Dan and Sasa populations, but the difference was only significant. Life cycle of Salamandra infraimmaculata along the southern border of its distribution. Picture 1 shows salamanders in terrestrial habitats. 2 and 3, salamanders migrating to breeding places. 4. Winter ponds and breeding places. 5. Female salamanders reaching the breeding place. 6 and 7. Salamander oviposition. 8. Sexual behavior and mating of salamanders. 9, 10, and 11. Larvae of salamanders. 12. Salamanders undergoing metamorphosis. 13. Juvenile salamanders moving from breeding places to terrestrial life. 14. Adult salamanders in a hole in the ground that serves as a hiding place. Salamanders in terrestrial habitats. Salamanders migrating to breeding places. In the first light rains in the fall, the salamanders, both males and females, leave the hiding places where they spent the summer and migrate to water bodies to spawn. The males work with their body raised to the leaf with the help of the animal legs as shown in the film. The S. Infraimmaculata populations in northern Israel are located near breeding spots and isolated populations. The female salamanders arrive at the spawning area, enter a pool of water bodies, a pond, pit, or a stream and live spawn the tadpoles. Adapts to the water depth the salamander swims or stands during spawning. In one pool in the western Galilee, normal pool, on a rainy day 8 salamanders spawn. Salamanders spawn live tadpoles wrapped in an embryonic membrane that falls off immediately after spawning. Differences between the male and female fire salamander. Cloaca of the male with spermatophores, cloaca of the female spermatophore. The spermatophore is located below the male and female. Stages of sexual behavior of the Salamandra infraimmaculata. 1. The male in a position of observing the female. 2. The male chases the female. 3. The male gets under the female. 4. The male crawls under the female until his head is under hers. 5. The male grasps the female. 6. The male and the female move their heads and tails alternatively. 7. The male stops moving and lays spermatophore. 8. The male moves and the female collects the sperm sac into the cloaca. 9. A photograph showing what is depicted in drawing 3. 10. A photograph showing what is depicted in drawing 8. Sexual behavior and fertility. A female moves to the spawning site. Sexual behavior and fertility. The female leaves the pond after spawning. After the migration to the spawning places, before spawning, and usually after a period in which the sexual behavior occurs, the salamanders return to the hiding places and live alone until the spawning season of the next years. Spawning of larvae in different habitats. Salamanders are spawning. Some of their larvae are born enveloped in an embryonic membrane, and others are born without this membrane. Salamanders of a subspecies in Europe are born alive. In the Pyrenees Mountains of Spain, a species whose larvae are spawned at a very advanced stage are found, even after metamorphosis. Some researchers believe that for all amphibians in the Urodilo order, the size of the clutch depends on body size. Activity takes place under conditions of 0 to 20 degrees Celsius and 60 to 100% RH. Larvae were studied in different types of aquatic systems: permanent spring, a rock pool, and temporary ponds. Growth pattern changed according to the pond types. Temporary ponds larvae mature more rapidly than in permanent pool or springs. There is also no difference in growth in the different areas, but there are differences in larvae growth between the various breeding places according to ecological conditions. In streams, as in Teldan, or in springs where water temperatures were lower compared to winter pools, the growth rate was lower. And the periods of larvae growth and complete metamorphosis were longer. A. Larvae inside the stomach of cannibalistic larvae of S. Infraimmaculata from the Hamama spring. B. Cannibalistic larvae and the larvae eaten by them from the Hamama spring. C. Large larvae with small larvae partial cannibalism. Damage to Salamandra infraimmaculata populations by human activity in creating water pits is a death trap in semi-arid habitats. The different breeding sites of Salamandra infraimmaculata on the southern border of its distribution were examined and mapped in order to estimate the damage caused by water holes that could represent death traps for salamanders in xeric habitats. Among the various types of breeding sites, the larvae of salamanders were detected in high numbers in springs and streams and water holes, and in relatively low numbers in winter pools. 2 water holes were examined in detail to estimate the damage caused to S. Infraimmaculata where breeding places are limited. A map of water bodies of potential breeding sites of S. Infraimmaculata according to the maps published by the government of Israel during the years 1941 to 2018 in the area of distribution of this species in Israel. Blue represents winter reservoirs, red represents springs and streams, black represents water holes, and yellow represents winter pools. The source of the map of water bodies of potential breeding sites was provided by the Tel Hai Map Archive. During the winter, both males and females enter a water hole for breeding. The percentage of males in and around the water hole was 42%, and that of females 29%. After the salamander's oviposition in the water, they try to return to terrestrial habitats, but are unable to do so and lose weight and die. The body mass index of salamanders decreases from winter after they move to the water hole to spring, when they are found in the water hole. The estimation of potential damage is about 300 mature salamanders annually. Salamander breeding site RPH1 from outside and closer to the inside. Entrance to salamander breeding site RPH3 from outside and inside. Salamander specimens in hiding between stones dried up during summer 2002. Karahizer and Demersoy discovered that Salamandra infraimmaculata have a karyotype of 24 chromosomes. For comparison, salamanders in Israel were taken from the Carmel. These researchers described for the first time the structure of the chromosomal system, the karyotype, of the fire salamander in Israel. They found that the fire salamander has 12 pairs of chromosomes. Genetic variation in Salamandra infraimmaculata from different habitats. Cluster analysis. The study utilized hierarchical cluster analysis to group S. Infraimmaculata individuals based on genetic similarities. Cluster 5 was found to be distinct from all other clusters, including the ST1 population, which was expected to be more predictable. Assignment of individuals to genetic clusters, displayed by color. Each vertical line represents the fractional membership of an individual to each genetic cluster. Horizontal axis represents sampled populations. Dendrogram from hierarchical cluster analysis shows grouping of S. Infraimmaculata Degani et al. 2014. Gene flow. The study assessed gene flow between clusters. Clusters 3 and 4 were identified as both sources of migrants and recipients of gene flow, suggesting genetic exchange between these clusters. Clusters 1 and 2 were also considered potential sources of migrants but received less gene flow. Phylogenetic analysis. A phylogenetic analysis, based on Ney's genetic distance, revealed that the Teldan population is distinct within its subpopulation, indicating genetic divergence within this group. Degani et al. 1014. More information about the salamanders at the southern border of the distribution appears in the free online books.